In this video of the cell cycle, you're going to learn the role that the cell cycle plays in a variety of activities. It's not as easy to develop and use a model to explain the role of cellular reproduction unless you understand where some of the components come from. What happens while they're developing, how it differentiates, how it divides, and then how it regulates itself, and then of course when the regulation goes awry. So how does a lot of this happen anyway? How do we get from inside the womb to a crying baby to a strapping young man? To become a human, it all begins with a fertilized egg. Once the sperm fertilizes the egg, that fertilized egg, which is called a zygote, it goes through cell division repeatedly. Cell division is through the process of mitosis, and that process gives rise to many identical cells that are all the same. But then these cells have to go through differentiation. That's a process that creates these special structures and functions that make each cell or variety of cells unique. Specialized cells become things that are different from each other: organs, tissues, whole organ systems. So you get half of the genetic material from your father and half the genetic material from your mother. They create a fertilized egg. There you are as a baby zygote, and eventually you will become an embryo through many, many cellular divisions and specializations. There's a lot of cell division going on when just the first 6 days after fertilization. An oocyte is released from ovaries. This is an ovary. In fact, this is a diagram of a woman's uterus. This is the other ovary. An egg is released into the fallopian tubes, and high up in the fallopian tubes is where fertilization occurs. That fertilization is where an egg nucleus and a sperm nucleus will combine, because you got half of your set of DNA there and your other half of your set of DNA there. So you need a full set of DNA. And when it combines to form one complete set of DNA, we have a zygote. Once we have a zygote, this cell will start its division, where all the cells are getting the same copies of genetic material for 30 hours or so. It'll go through stages where it divides from 1 cell to 2, the 2 cells to 4, the 4 cells to 8. And by the time we get to 72 hours, we become a morula. The morula is still a cluster of cells together, but after about 4 days, our cells start to migrate towards the ends of the fertilized ovum, and this is where we start to see differentiation. After about 6 days, a blastocyst is formed, and a blastocyst no longer needs this external barrier to contain the cells. It has now got highly differentiated cells that are forming the very first coelom, is what that's called, and that will be your internal body structure. The part of your body that will ultimately end up being a coelom, or a mouth opening and an anus exit. Now, perhaps you've heard a lot about stem cells in the news, and stem cells are undifferentiated cells. They haven't decided what part of that blastula they're going to be just yet and where they're going around your coelom. They can still become differentiated into one or more different types of specialized cells. There's really 2 different types of stem cells. There's embryonic stem cells, and then there's adult stem cells. Embryonic ones are cells that were never really differentiated, hence from an embryo. And adult stem cells are found somewhere in the adult, usually the bone marrow, where they're only kind of partially differentiated, and they can become bone and blood and cartilage. and fat, other types of condensed tissue. But embryonic stem cells are special in that you have a cluster of cells that haven't decided or been programmed on what they're going to do just yet. They can become bone or heart, even liver, part of your intestines, or even brain cells. Now, remember that fertilized zygote that eventually became a blastula where an opening started to form? Once that opening starts to form, we start to get the first differentiation, and that is when some of the cells decide they're going to be an ectoderm, the mesoderm, or the endoderm. The ectoderm are things that are going to become on the outside of your body, like skin cells. The mesoderm, or middle, are things that are going to create ultimately your blood cells, your smooth muscle cells, skeletal muscles. And the endoderm are things that are going to be forming on the inside of those, inside of the mesoderm. They'll become things like stomach cells, lung cells, pancreatic cells. So the first line of differentiation and specialization that we see is really that blastula turning into a gastrula and forming the three ultimate layers of what are eventually going to be a little human one day. So an implanted blastocyst becomes a gastrula, and it's an embryo with three differentiated germ layers. Organogenesis is the process of which the body organ and organ systems form following gastrulation. Gastrulation, the differentiated and specialization of a gastrula, which has ultimately three layers. From there, cell differentiation just takes off. A cell will become the most basic unit of life that has all the characteristics of life inside of it. And every muscle cell contains all your DNA, if it's an eye muscle cell or a leg muscle cell. We're going to get lots of those cells working together and form a tissue. A tissue is, for the most common function, think of it more like muscle tissue. That's a layer almost of cells. Lots of tissues or sets of tissues working together for a common function form an organ. An organ would be like the stomach. Sets of organs working together for a common function form an organ system. So for example, you have an esophagus, and a stomach, and intestines. Multiple organs working together form your digestive system. When you have a lot of systems working together, we get an individual called an organism. We can contribute all of this to the cell cycle. It's a repeated pattern of growth, DNA duplication, and cellular division that occurs in every single eukaryotic cell. Its purpose is to grow cells and repair them. The cell cycle consists of three main phases: interphase, which is cell growth, mitosis, which is cellular division, and cytokinesis, which is splitting of the cytoplasm. All of this is interphase. It consists of G1, S, and G2. Then the cell will go through its division, which is mitosis. And then the very last part is cytokinesis, where it splits its cytoplasm. Interphase, or the growth phase of the cell cycle, is when the cell is carrying on its normal function and doing its thing, and it's growing. It's divided into three relatively even separate phases. G1, also known as gap 1 phase, is cellular growth, and at that time, the cell is making proteins. S phase, also known as synthesis phase, is when DNA replication is occurring. Cell's not dividing yet, but it's getting ready to. So it's replicating its DNA. It's doubling the number of chromosomes that are inside of it. And then G2 phase, or gap 2 phase, we have more growth, more protein synthesis going on. It's growing because it's eventually going to have to split its cytoplasm. So it's got to make more of it to give away to the new formed cell. At the end of interphase, the cell is going to have 2 full sets of chromosomes because it's about to ready to duplicate. And when it duplicates, it's going to make 2 cells, and it has to make them equal.